Okay, so thank you very much, Ola, for these fantastic presentations, and I hope that the CF Trikafta not only will be available for CF lung patients, but also for pancreatitic patients. So our next speaker will be Martin Chalfie, and let me a little bit introduce of his background and his story, actually. And plenty of times people are asking whether scientists are only in the lab, they are only interested in mechanisms, and it's not true. I mean, scientists are really like any other people. They are interested in so many things. He was a really brilliant classical guitar player as well. But in his background, what I must say, what was really very impressive about the interest of different things in their entire life. He, as a child, he already had microscope and chemistry set, so he always wanted to find really new ways. So his first experimental steps after he went through, he entered Harvard University. He finished it, and however, he had not really huge confidence in his ability to do research. And actually, finally, he had so many failure in his experiments, so he stopped doing science and left the field. And he went to work for plenty of works in factories and other part. And one job which came to him finally was actually again in the lab, when he returned to a lab, as I think in a summer part. And after he got again inspired by that to start really again in science. And after that, he actually moved to Yale in 71 and started doing that and did his PhD actually with Robert Pullman in 1977. And after his work was continuously in science, and he was working also with Sidney Brenner and John Sulston. And in 82, he moved to Columbia University in New York, and he is still working actually there. And if you look at what was his discoveries, and I come to the worldwide known 94 papers, which was published in Science for the very first time in the world, he could prove that the GFP, the green fluorescence protein, is not harmful. It's a small protein in their cells, and they can use not only like actually you can see like different cell lines or yeast, but also for animals. And plenty of people got inspiration when they looked at these green animals to do different other studies. And of course, in the field, he had lots of competitors. And actually, I also mentioned to him that I will introduce one of the main competitors, which he wanted actually. He showed that GFP is not harmful, but he wanted something which is the other competitors, which is not harmful, but also fun. And his biggest competitor was William Steig. I don't know you know this person. He published his data in 1990, 4 years before you. But at that time, it was actually forgotten. But I had a contact with the Nobel office in Stockholm, because his recordings were actually discussed before the Nobel meeting. And if I ask to start it, you can realize that other developments could have done, and this was, the video recordings were published in 2001, about in a much better model, So this animal model was even more, than a normal animal. However, they could, make feelings in people, scary people, even could marry, in this animal. However, because William Steig couldn't show the cDNA of this model, actually, finally, it's not surprising that this green animal model was not as famous as in science, like Marty's models, and I think that the model he provided was over 10,000 times cited, and hundreds of thousands people used actually for further years. So it's not surprising that he received the Nobel Prize with Osamu Shimomura and Roger Tseng for discovering the gene fluorescence protein. By myself, I used several times, and I think nowadays in basic science, people are tremendously using. His wife also a really brilliant scientist. I also looked at the net, and she has really brilliant papers, and they are together working. And of course, he received huge amount of honors. And not only, you know, the Nobel Prize, but even after Nobel Prize, he received one. He also became to be a foreign member of the Royal Society. And this is for everybody again. If you do science, you not only do for yourself, so almost you don't do yourself, you do it for the society. And when you reach a level, you have a chance to do something for the future. Many scientists are unwilling to spend their very valuable time on important things, which is not clearly inside there. But also Marty is really one of the driver of the climate change part, and not only the climate change, but he's also an ambassador for human rights, and he had unbelievable lot of activities inside the field. So it's really a huge appreciation, and I admire that he spent huge amount of things which is in the 21st century highly important. And of course, as I mentioned at the beginning, people, you know, not only interested in science, but any other things. They are the same persons like we are. He is very much interested in playing music and also puzzles, stems. They are one of his best part. And he also have wisdom for the young, because we are at the Translational Center, but also in my journey, what I realized, to translate something, you need to do basic research first. So you first have to understand what's going on, after you need to further develop, and the last time when you need to actually translate back. So this is the very first time for Marty being in Hungary, and I really would like to highly appreciate and highly thank for you to coming. We spent 1.5 days so far. I enjoyed every minute. We already got plenty of ideas how to go forward with the program. So thank you very much for your incredible time. And we hope that this is not the last time that you are in Hungary. And we wish you all the best, and enjoy the forthcoming date. And we just can't wait to listen to your presentation. Thank you very much, Marty.
Well, thank you. That was quite the introduction. I think that proves that some people should never be allowed on the internet. But thank you, Peter, very much. So the title of my talk, I'm going to give a very general talk, talking about ideas in science. One thing you didn't mention is that before I got my PhD, when I couldn't figure out what else I should do in the world, for 2 years I was a high school teacher. And that was, I think, very formative for me, frankly. Anyway, let me talk about, I've titled this talk, The Continuing Need for Useless Knowledge. And this is actually a steal of a very interesting article. I hope this works, and I figured it out, by this man, Abraham Flexner. Abraham Flexner is sort of known in the United States, I think, for two things. One, he was in charge of a committee in the early 1900s called the Flexner Committee, in which they laid down the rules of what every pre-medical student had to take as an undergraduate, all the chemistry and the math and the biology and the physics. So he has ruined the lives of medical students or pre-medical for over a century, and we still do that now. But the other thing he was the first person to come up with the idea and the first director of the Princeton Institute for Advanced Study. And in 1939, he wrote this article in Harper's Magazine, which he entitled The Usefulness of Useless Knowledge. And you can see the quote at the bottom. He says, intellectual life is on the surface a useless form of activity, but it is unexpectedly the source from which undreamed of utility is derived. And he starts the article off in a very interesting way. Remember, it's 1939. So he's talking, let me get to the right slide. He's talking to an industrialist, George Eastman Kodak film maker, very important industrialist. And he asks him, who is the most important scientist in the world? And Eastman immediately has an answer. He says that it's Marconi, because Marconi has invented the radio and wireless, and that's really phenomenal. And basically, Flexner writes, I told him that he was completely inevitable. He's not important at all. What is really important for the development of radio, that somebody else would have done it if Marconi hadn't done it, because the really important people in all of this were James Clerk Maxwell and Heinrich Hertz, because these are the people that provided the understanding of electromagnetic waves. And without that understanding, there would be no radio, there would be no wireless. So he says that you need that new knowledge. I would say that you need both new knowledge and new applications. It's really both things that are needed. But his point is there's a lot behind any advance that goes on in science. And I'm going to give you a couple of other examples that I've found over the years that I enjoy. One of them is Mark Twain, who in a letter to Helen Keller said 1,000 men invent a telegraph or a steam engine or phonograph or telephone or any other important item, and the last man gets the credit and we forget all the others. And to drive this home a little bit more, I'm going to show you an infographic. I'm also going to warn you that you will not be able to read the infographic because it's quite large. But if you see at the bottom of the slide, you can see that it was by the Quartzoff Company, and you can go to their website and you can see this. Because what this is, if we look at the top of the infographic, it is, how did we get the iPhone? How did we get a smartphone? With its Wi-Fi and telecommunications ability, camera, all the other things, the computer in it, everything. What was needed? And as you go down the infographic to a slightly lower place, you find all these scientists were needed and all these discoveries and applications were needed before you could get to the iPhone. And that's the top of this chart. If you go to the bottom of the chart, you find that there's the discovery of the first lens, or the discovery of zero, or using binary. Just all the things that were needed to accumulate to be able to produce this thing. Without this basic research, without the useless information, none of this is possible. So it really is building on things one after the other. And I'm going to show you my favorite video. I had nothing to do with this video. It was from a contest in 2014 that the Federation of American Societies of Experimental Biology had. They said, please make us a video to show the importance of basic medicine, or basic science rather, for medicine. And a group of graduate students and postdocs at the University of California in San Francisco made this video. So I'm going to show it. I hope the sound is on. We'll have to see. Nothing happened then.
It's a sunny day in San Francisco. We set up a table by the water and asked people this question. If it's the year 1960 and you had $10 to give to science, would you spend it on A, developing an affordable treatment for diabetes, or B, figuring out how bacteria protect themselves? The majority of people we surveyed wanted to spend their money on a diabetes treatment, but what really happened 50 years ago? The government decided to spend money on figuring out how bacteria protect themselves against viruses. The National Science Foundation, the NSF, and the The National Institutes of Health, NIH, are two government agencies that allocate tax dollars to scientists. And they decided to fund groups of researchers who thought that studying how microbes protect themselves was an interesting question. At the time, there was no way of knowing how this basic research could affect human health. They were simply curious about understanding something about bacteria biology. So what did the scientists find? Normally, when a virus enters a bacteria, it adds its own DNA to that of the bacteria. In doing so, the virus takes over the bacteria and uses it to make more viruses. Scientists found that bacteria protect themselves from viruses by selectively cutting the viral DNA. This basic finding led to a scientific revolution. Scientists learned how to cut specific chunks of DNA, and they could use this technique to move genes around. They could even move human DNA into bacteria. And because bacteria are very good at making more of themselves, scientists can use them to make large quantities of human proteins from the human gene that they introduced. So why was this technique useful for modern medicine? Well, one breakthrough was the efficient production of human insulin by introducing the human insulin gene into bacteria. This was a very significant advancement in diabetes treatment. Before using bacteria to produce large quantities of human insulin, insulin was expensive, unsafe, and less effective because it was harvested from fetal cows. In fact, the first person to get an injection of cow insulin suffered from a severe allergic reaction. Because of a long line of federally funded basic research, human insulin is now an affordable treatment for diabetes. By asking how bacteria protect themselves, the scientists discovered that bacteria can cut DNA. This discovery was developed into a basic technique used in thousands of labs around the world to create new knowledge and to ask new questions that have led to all kinds of therapies and technologies, including insulin, DNA fingerprinting and biofuels, the human genome, growth hormones, cancer drugs, stroke medication, vaccines, HIV meds, antibiotics, and many, many more. Given enough time and funding, who knows what is to come? Many other health benefits and current discoveries would have been nearly impossible without federal funding for all kinds of basic research. Basic research is an investment in our future. It broadens our understanding of the world we live in, and the potential benefits are limitless. Unfortunately, science funding in the US has plateaued, decreasing purchasing power and significantly hurting our competitiveness and innovation. You may not know it, but you and your tax dollars are part of the discovery process. Thanks to you, scientists discovered that bacteria can cut DNA. And thanks to you, insulin, other drugs and diagnostic tools have been developed and are helping patients.
So thank you for supporting basic research.
We are the scientists who depend on you, and we are excited for the future that lies ahead.
Pretty great, isn't it? It's absolutely fantastic. Anyway, question, so there's an award that was started in the United States a few years ago called the Golden Goose Award. And it's specifically for research, as it says, for scientific studies or research that seem obscure, sound funny, the results are totally unseen at the outset, but which ultimately led to major breakthroughs that had significant societal impact. And it's very nice to be one of the first groups of people that were considered for this. So they gave it for green fluorescent protein, the invention of maser, and the discovery that coral, when cleaned up, is a spectacular substitute for bone. Other things that they've done, and my favorite of this, which I'm going to spend a minute telling you about, is rat and infant massage. Because there were a group of researchers that got this award, and what they were doing, they started off an experiment. They made a genetically modified rat that they wanted to study some process, but they were very, very concerned, because of all the work they put into it, that the mothers might kill the babies. So they decided, okay, we better take the pups away from the mothers as soon as we can. And they did. And the pups didn't thrive at all. It was a disaster. If they left the pups with the mothers, they did fine. So they kept them with the mothers, But then they asked the question, what were the mothers doing that was helping the babies? It wasn't nutrients or anything. What was happening? And they observed the mothers, and they found that what the mothers were doing was quite often licking the babies. So they decided to do an experiment. They didn't lick the babies themselves, but they took a swab and they stroked the babies. They gave it tactile stimulation, but away from the mother, and they thrived. And they realized that all that licking was stimulating mechanoreceptors or whatever that were very necessary for the babies. Well, this is nice. They published it. But what it led to was a realization that at premature baby wards in hospitals, maybe there should also be more tactile stimulation and more stroking. And it has apparently gone all over the world and is used in neonatal units. So an observation can lead to something quite spectacular that affects the entire world. Let me go on from here. I think we need the idea that new knowledge yields new approaches. New approaches are the things we need to get. So the real question is, how do we get them? I think after years of trying to talk to patient groups and so on, people, I think, have an understanding in medicine that you can't just go after the disease, you have to go after the basic science underlying that and gaining some information for that. And I think that's a good thing. So it goes back to that line that you quoted from me that if you need, and that other people have said as well, if you want to do translational research, you need to have something to translate. These advances, as I say, require the basic knowledge, and we still need much more of that basic knowledge. But I'm going to expand this away from medicine. I think this problem is much more general, and I'm going to give one example. I have a good friend at Stanford, Manu Prakash, is an unbelievable educator and inventor. If you've heard of his name at all, you've probably heard of it because he's the guy that invented the paper microscope called the Foldscope that costs, it's a whole microscope that costs $1.50. He sent over 2 million of these all over the world. It's a spectacular thing. But one of the things he's interested in now is climate change. And I don't know how it is described here, but in the United States, almost everything about climate change is an engineering problem. How do we mitigate the rise in temperature? How do we get rid of the carbon? It's viewed as an engineering problem. And what Manu has realized is that we have no idea how the oceans serve as a repository for carbon and the microbes within them. We don't know the basic science of how the planet works. And until we understand that as well, mitigation things are not going to be enough. And so again, even here, we need to understand about basic research. So how do we do it? There's a wonderful quote by the physicist Enrico Fermi that goes, if the results confirm the hypothesis, then you've made a measurement. But if the results are contrary to the hypothesis, then you've made a discovery. And this has been seen many times, and I'm going to give you some examples from the Nobels. So the first are Robert Wilson and Arno Penzias. They were at Bell Labs in New Jersey. Robert Wilson was very concerned about redoing an experiment that had completely failed during his PhD. I think he was feeling a little guilty about that. And he and Arno discovered that somebody else at Bell Labs had gotten the company, the place to build this 30-foot microwave telescope. And they said, oh, we can use that for our experiments. But before they could do the experiments, they had to calibrate the machine. So they pointed at the Milky Way, and they get a beautiful microwave signal. it works great. It picked that up. And then they zero it out by pointing it away from the Milky Way. And it doesn't go to zero. And they can't figure out where this residual activity is that they're measuring, and they know they can't do their experiment until they get rid of it. So first they think it's pigeons that might have roosted there, or electrical lines, or something else. They eliminate everything, and then realize that they're actually measuring something. They are measuring the background radiation in the cosmos, which was the first indication of the Big Bang. And that's what they got their Nobel Prize for. So they didn't get their Nobel Prize for doing an experiment. They got their Nobel Prize for failing to calibrate their machine, which is pretty interesting. The other person I want to say is John O'Keefe, who discovered the place cells. And if you read his Nobel Prize speech, he says that he was doing electrophysiology by sticking his electrodes in, I think, the thalamus, when one day, by mistake, he stuck it in the wrong part of the brain and discovered these really interesting cells. So a complete accident. And the third example is closer to my heart, and that is Osamu Shimomura. Osamu Shimomura is an absolutely was an amazing person. He died in 2018. He's written a memoir, which I recommend highly to everyone. But he was trying to figure out a fundamental biological problem. How is it that this particular jellyfish, Aquaria victoria, produces light? Fireflies, glowworms, bacteria, fungi, many different fish, many different organisms produce light. And it's a really interesting question. What is the chemical reaction that can actually release enough energy as light? And how is that done? And every species does it differently. So knowing what fireflies do don't tell you anything about any other organism, for the most part. So he was interested in this organism. He was working at Friday Harbor Labs in Washington State in the United States, and he's grinding up hundreds and hundreds of jellyfish, trying to isolate the biochemical entity that's producing light. And he's failing all the time. Finally, one evening, he has done the experiment for the umpteenth time. It has failed again. So he says, I'm going home, I want to have dinner, it's dark outside already. And he takes all his samples and he throws them away in the sink, which happened to have some seawater and probably jellyfish parts and other things. Turns off the light and is just about to leave when he looks back at the sink and it's glowing brightly, something he had never seen before. And that stops him. I should say this is the real scientific method. If you throw it in the sink, the floor, the desktop, usually works. In any case, he thinks about it and says, there must have been something in the seawater. What was in the seawater that I've never added to the preparation? And he thinks about that for a while and says, well, there's calcium there. And he goes back, does the prep after a couple of days, and sure enough, every time he squirts in some calcium, he gets a flash of light. And he's able to use that flash of light to purify the protein, which he names after the jellyfish. And so this bioluminescent protein, a protein that produces light, he calls a quarin. And a quarin plus calcium produces light. And then I'm sure he looked at that and went, oh no, this is horrible because the jellyfish is green, but this light is blue. I've got the wrong color. He adds calcium to all the other samples, no green light gets produced. And he thinks about that for a while. So that's the second accident. He thinks about that for a while and says, wait a minute, maybe there's something that converts the blue light to green. And so he takes a handheld lamp, doesn't need calcium, doesn't need a quarin, just blue light, and he shines that on all the samples, and a completely different sample does have a protein in it that the blue light can activate to get green. He writes about this in the purification paper of 1962, as a footnote, footnote number 3. As far as I know, it is the only footnote that won its writer a Nobel Prize. Because what he says in it is there's this other protein called the green protein, in which we now, of course, call the green fluorescent protein. And it is a corrin plus calcium that does produce green light, but instead of making green light, if GFP is around, you get, instead of blue light, you get green light. And I heard about this, not in 1962, 1989. I was listening to a seminar and I heard about this, and I realized what a wonderful tool this would be, because the molecule, all you need is blue light in, you get green light out. I would also point out, this is the molecular structure of green fluorescent protein. This, as far as I know, is the only molecule that looks like it should, right? Water doesn't look like water. You know, carbon dioxide doesn't, you know, none of this looks exactly like it should. It's a lantern. And it's astonishing that this molecule looks the way it is. And I realized if you put this into cells, you wouldn't have to add anything to see it except for blue light. You could see wherever it was because of the green light that was produced. At the time, I was doing genetic studies on a small worm that was transparent. So I was overjoyed about this. And eventually, a graduate student, Gia Skirkin, and a collaborator who got the DNA for this, Douglas Fresher, and I actually were able to put this together and put it into these worms that we were studying, or first into bacteria, and show that it could work there on its own, and then into the worms, and so we could actually now see the nerve cells we had been studying in living animals. And so it was very exciting, and then all of a sudden a lot of other people started putting them in all sorts of organisms and looking at cells, and it's a small molecule, you can see everything. The nice thing about GFP, to give a little advertisement, is because it doesn't hurt cells for the most part, unless you overproduce it way too much. And all you need is to shine blue light on it, which is fairly innocuous to cells. You can watch living processes go on. I want to show you two movies that were made by someone I've never met, Rosalind Silverman Gavrilla. I should point out that I have a color code on my slides. Everybody that's worked in my lab, their name is in red. Everyone who is a collaborator, like Douglas Fresher, their name is in blue. Everyone that did something I wish I had done, their name is in black. So if you see here, what you see on the left-hand side is GFP attached to the tubulin, part of the spindle process that's going to be made in cell division. And she made this movie that shows 4 rounds of nuclear division in an embryo in fruit flies. And so it's quite sped up, but when you look at it, as I hope will happen now, you will see the spindles form, and then the cells, spindles form again. And I think what strikes you, and struck her because she called this movie In Synchrony, is all of these things are doing at the same time. And it immediately introduces the question of how in the world does that happen? And we know quite a lot about this. I think the past thing, Tim Hunt has been here, so we know about the work that he has done. On the right-hand side is a falsely colored image. It's still GFP, but the colors come about by the amount of material that's there. And the convention is to use the rainbow. If you start at the blue, you have the least. As you go finally to the red, you have the most. And so this is a falsely colored thing. And here GFP has been attached to a small peptide called a nuclear localization signal. So if there's a nucleus, it goes there. And when the nuclei break down for the division, then it's going to go all over the cell. So we'll watch the same thing. And here, the nuclei break down. It's all over the embryo. The nuclei are going to reform, and the stuff is going to get sucked back into it. She names this after the Van Gogh painting, Starry Night. And What you see here is it's not synchronous. It's going as a wave from the lower right to the upper left-hand corner. And people that are more knowledgeable than I about this have suggested maybe her thumb was a little heavy on the prep. But this shows how sensitive this can be to be able to watch things in living organisms. Lots of different things have been done, thousands of experiments that have led to more fundamental knowledge. One example I quite like is the discovery of phase-separated particles in the cytoplasm, which is one of the most phenomenal new discoveries that came about by looking at GFP by Cliff Brangwyn and Tony Hyman, and as an example, but it can also be used for applied work. To ask a question, for example, how is the HIV virus passed from one cell to another? Is it like the textbook example of viruses, that cells explode and spew out their virus and so it's available to everything, or is it somewhere different? And people in 2009 made a GFP version of HIV, infected this mouse cell, and you can see it infecting a nearby cell here, just any second there. There it goes into that cell. No explosion. So if you want to control it, you got to really think about a different way of interfering with this transfer of virus from one cell to another. So there can be applied, there can be fundamental research. I think that it's not that fundamental goes to applied, it's really cyclical. It's fundamental gives rise to applied, which gives rise to more fundamental, and on and on. But there's another aspect to this as well, and that is the unexpected. And there has been a tremendous amount of unexpected. Let me define unexpected. Things I never imagined that people did. So some examples of that. There is a group of scientists in Japan who decided to put GFP and some of the other color fluorescent proteins into silk moths, and they made fluorescent clothing out of the silk for this. I'm not sure this is exceptionally useful, because I think if you want to show off your neat clothing, you have to always carry a UV lamp with you and walk around with it as a little, or just go to discos. I don't know if people do that anymore. Anyway, that's one example. Other people made a laser, a cellular laser, using GFP as one of the components. Bob Berladge at Oak Ridge National Lab put GFP under the control of a promoter that was turned on by the explosive TNT into E. coli. So if there was TNT in the culture medium, the coli would produce green. And he did that because he wanted to have a way of detecting landmines. And so he had the ability to have someone bury 5 landmines in a 3-meter by 5-meter plot of land. They were disconnected, but they did have TNT, and it's known that landmines leak TNT. He sprayed the area with the bacteria, came back at night with the UV lamp, and took this picture. There's been a number of other groups that have been trying to do this. Landmines are one of the absolute worst elements of war because they persist, and they maim innocent people probably more than combatants. And this is still something that's being worked on, in part because you don't want to have any false negatives. And trying to make sure that there are no false negatives is a very important part of this. So I know of at least 3 or 4 groups that are working on this. Another unusual use of GFP, Eduardo Carc is a Brazilian artist. He lives in Chicago in the United States, and he arranged for a French biotech company to make Alba, his GFP bunny, to express GFP in its skin. And he used Alba, who was the family pet and lived a normal, apparently happy life. He would bring Alba to art shows to get people to talk about the connection between science and art and technology and art. and so on. And then the last, which is not the one you gave, but another example of this, is the first purported human transgenic with GFP, and that's this person from Ang Lee's movie Hulk, and it actually starts off with the jellyfish, bobbing away, and the hypodermic comes in and sucks out the GFP, which obviously got into Bruce Banner, and you get the Hulk whenever he's mad, expressing GFP. So I actually know the screenwriter for this. Our daughters went to school together, and I asked him, I said, my lab made me go and see your movie. I really liked the beginning. How did you know about my work? And he looked at me and said, I have no idea what you work on. And I said, no, that's the GFP. And he said, we had a student from MIT working on the set, and he made us do that. Fine with me. It was very good. But lots of different things. There have been lots of different tools that have been made of it that have been really quite wonderful. But I want to go on from that. If we say that most discoveries are accidental, how can we increase serendipity? That's really the question. How do we get more of these discoveries? So I'm going to talk a little bit about that. I think there's two parts of it. One is to question what is right in front of us, and the second is to question our assumptions. And I think these are both remarkably powerful ways of addressing things. So I want to talk first about questions right in front of us. And I asked this of people yesterday when I talked, and hardly anyone answered, so we'll try you guys. I worked, my first scientific paper was working on the major transparent part of the body. If you know what it is, don't say anything, but just raise your hand. If you know what the transparent part of your body is, raise your hand. Yeah, that's what usually happens. Hardly anyone does. So now I'm going to give you all a hint. It's right in front of your eyes. Because it's the cornea. But even though I have not met many people that will answer the question, what is the transparent part of your body? And you could say the lens and other things too. But I have not yet met anyone, other than the people I happen to be lucky enough to work with for this first paper that I had, who asked themselves the question, why is that tissue clear? Why can we see through it? What makes that different from all the other skin and surface material that we have? What allows us to see? So literally a question that has been in front of ourselves our entire lives that I would think the vast majority of people do not look at. And there's a lot others, but I'm not going to give those examples now, but I'm going to tell you about my second cousin Ray. Because Ray is a professor of physics in Cambridge, England. He's a spectacular member of the Royal Society. He does spectacular work. I only learned he was a cousin about 4 or 5 years ago. So I'm very proud now that I have this guy as a cousin. Because what he does is almost all of his research is based on those same questions of what's in front of us. So, for example, he was studying how fibers in a bundle all work together, how they interact, what happens to those bundles. But being Ray, he didn't write the paper to say scientific discoveries of how bundles of fibers come. No, he wrote a paper that described what determined the shape of a ponytail. And for that, he won the Ig Nobel Prize in 2012. Other questions he's worked on: why are icicles shaped the way they are? If you look carefully at any of your clothes, they're actually made up of very, very tiny fibers. And so he's trying to figure out why our clothes don't just fall apart, fall off of us. He's had a paper on that. And then last year he had a paper not on why do cicadas come out in 17-year periods, but why do they come out all at the same time? And he and his wife, who's a fantastic mathematician, actually figured out that there must be some signaling device that allows all the insects to emerge together. So it's just a fascinating thing. All of these very obvious questions, all sitting there for somebody to look at. I've talked to Ray about whether he and I can collaborate on a project together of why it is when you put your shirt in the washing machine, it always comes out inside out, but we're working on that one. But some other things. I've been very fortunate to be invited to what's called the ISEF, the International Science and Engineering Fair, which is a culminating science fair for high school students from all over the world. And one day I was there, and there was this high school student, and I stopped by her poster, and I wanted to find out what her work was. And she gave probably the most unusual introduction to a scientific talk I have ever heard in my life. What she said was, the first thing you should know about me, I'm a ballerina. I haven't heard that often. And her name is Abigail Fried, and she then told me that ballet performers, especially at the very highest professional level, have a real problem. And the problem is with their shoes, because the shoes have a cardboard sole, and after one performance, that sole is no good. So they have to throw the shoes away. And it's $110 or so every pair of shoes, so it's very expensive if they're performing at an incredibly high level. And so she said, so that was my project. A high school student. She said, I realized that there was a carbon composite that I could use, and so I tested whether 1, 3, or 5, actually she did a whole bunch of other things too, what gave the best flexibility. And she found that the optimum flexibility was 3 plies of this material, and it lasted for months. And I thought, what a perfect project, right? She sees a problem, she figures out how to address it, and she solves it. I was very impressed. It was fantastic. Anyway, let me go on from here to talk about the other aspect of this, and that's questioning our assumptions. Now, I'll give some examples. Peter said I like puzzles. I do like puzzles. And one set of puzzles is the matchstick puzzles, which you may have seen. For example, this one, you're given this pattern. It says, move 3 matches and just make 3 perfect squares. And you sit there and you swear at the matches and everything. But eventually you move them around, maybe, if you're lucky, and you get that pattern by moving 3 matches. And then the next problem is, here's a fish, move 3 matches and turn the fish around. And so again, you're sitting there and you're moving the matches, and then finally it may come to you that you can move them like that. And you can move the fish around. And then, after you've done that, the next problem, or could be the next problem, is this one. Make 4 equilateral triangles from 6 matchsticks. And you move them on the plane of the board, wherever you're working on, and it's really, really difficult until you suddenly realize that maybe it's your assumption that's wrong. Because no one said that you should only think in 2 dimensions. If you change that and start thinking in 3 dimensions, you get that. But there's actually another assumption here. That assumption is all the triangles have to be the same size, right? Why? Who told you that it had to be all the same size? It's, again, our assumption of what we're doing here. And you can actually take the 6 and almost on a plane make it such that you have 2 large and 2 small triangles in the set. And then, after you've gotten that, and then you get real cocky, okay, now I understand all this, then they throw you this problem, which I really quite like. Make these 3 matches into 6. You cannot break the matches. Anyone have an idea? You're going to kick yourselves. Yes, exactly. Why are you thinking about shapes? Could be a Roman numeral. So very different, but it's the assumptions. I have a good friend, Muming Pui, was a faculty member in my department. His entire career, which is an illustrious neurobiological career, has been built up on what he calls the retrospective approach. In this approach, he goes first and finds an interesting hypothesis in a textbook. For him, the textbook is a second edition of the cell biology text, the molecular biology of the cell. Then, when he sees something, he says, why did they say that? Why do we believe that? And he goes back and he looks in the literature, and he usually finds that it's based on really wild extrapolation. There really isn't good evidence for it. And he realizes he can now do the right experiments to test this hypothesis. So he does. He tests the hypothesis with modern methods. If he gets the same answer, of course, he's now confirmed it. So that's a paper, because now there's a strong basis for thinking this happens. But many times he finds out they were completely wrong, and that's also a nice paper because he's now discovered something new and has evidence for that. So he then publishes this and everything. And this is what he says he's been doing his entire scientific career, just questioning the basic assumptions in the field. So with that, I want to end with one of my favorite science quotes about the importance of basic research. This was done by Robert R. Wilson, different Robert Wilson from the one I talked about before. Robert Wilson was the physicist. He's also an architect. He's also a sculptor. He was also the first head of the Fermi Lab, which until the CERN Super Collider went on board was the largest particle accelerator in the world. And in 1969, which was during the time when the United States was fighting this war in Vietnam, he was asked to explain to Congress why this physics lab should be built. And he went there, and the committee in Congress went to him and said, okay, but you know, we're very concerned about national security. Tell us, how many ways will this particle accelerator help national security? And he looked at the senators and representatives and said, none. That was not the answer they wanted at all. So they asked him the question several times, finally culminating in the question, look, in what respect will this particle accelerator aid the national defense? And to this, Robert Wilson said, it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with, are we good painters, good sculptors, great poets? I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much.
Thank you so much, Professor Chalfie, for the insightful talk. I would like to open the floor for discussion. If anyone has a question to ask, please.
Okay, so good morning, everyone. First of all, I would like to express my gratitude for you to have this wonderful and experienced presentation. Okay, so like generally, I have two questions I would like to ask you. First of all, I know like there's a lot of like massive challenges when you apply the GPF to the living cells at the beginning. So I would like to know, like during those experiments or like those, when you're facing the challenges, what make you like keep your mind, like conceal yourself, like keep doing this research during these years? This is my first question. And secondly, because like programming scientist, so I know like as the rise of the AI model or like big data models recently, so a lot of like researchers, they would like to combine their research with the AI model or those like big data model. So even some researchers want to use some AI model to help them to summarize their content or even write the articles. So in this case, I would like to know like your attitude about this part. Thank you.
Okay, so the first question, and you're not going to like this answer. Before we worked on GFP, people knew about the molecule. It had been studied by Shimomura, and they had known the amino acid sequence. They knew that nothing needed to be there except the amino acid sequence. But what the protein did was so bizarre, they had no idea how that came about. So you'll know that standard proteins are a chain of amino acids, a linear collection of atoms that make up what's called the peptide backbone. GFP does something unbelievably strange. A 5-atom chain in the backbone cyclizes. So instead of getting a chain, you have a chain and a circle and a chain. That's absolutely needed for the function, and no one had any idea how it was going to be made. They thought, oh, there must be an enzyme that does that conversion. It has to be, so there's a converting enzyme, maybe 2, maybe 5, who knows how many were needed to make that protein. And so because of that need for a converting enzyme, everybody thought the experiment should not be done. Okay? It was stupid to do it. So they didn't want to do it. We decided, what the hell, let's try it. And that experiment worked the first time, because when we put it into into bacteria, we found you don't need anything other than itself. It is its own converting enzyme. And so it worked immediately. So there wasn't any agony. There was other agonies at other times. But you're asking a more general question. How do you deal with frustration? And I'm also going to say something horrible in that regard. And that is, I have spent most of my career looking at mutants that affect touch-sensing cells in the nematode Cinorhabditis elegans. And the collection of mutants actually gives us 2 sets of things to study. Some mutants are touch insensitive because they don't make the cell. Those genes are needed for the development, differentiation of the cell, and so they lead us into questions about how do you get specific cell types. Others, the cells look perfectly fine, but they don't work. And that leads us into a completely different question of sensory biology. How does a cell respond to mechanical stimuli? Sometimes one works, sometimes the other works. So my suggestion is, if you're frustrated, do twice as much work. Another way of putting this is, if you look around a lab, an individual working in the lab might be unhappy, but if the lab is any size, the head of the lab is always happy because somebody's work is working. So you need to have some, do something else. Nobody likes to hear, if you're upset about things, do more. But that's what's worked for me. The other question was?
Like the one for the AI.
Oh, AI. So I think there's a lot of fear about AI and a lot of concern. I'll tell a couple of weird stories. I'm sort of an old guy. So I have to go periodically to the doctor and get a colonoscopy. And when I went to the doctor this time, he said to me, I just want to tell you that while I'm doing it, we also have an AI program that's also reading the output. And I almost got up, couldn't get up, but I almost got up and cheered, because who wouldn't want a second opinion? Who wouldn't want something to say to the doctor, hey, look over here, is that something? And have him decide whether it's there or not. So to me, the idea of using AI as an assistant is really terrific. In the United States, I don't know about here, but in the United States, the way AI is described in the news all the time, it's basically part of the overall and continuing generational problem of our kids are horrible, right? Because kids will cheat. They're going to cheat in school. They're going to do this. And this is the thing. So, and especially they're going to have ChatGPT write their essays for them. The big fear. So I did that. My name is unique. There is no, as far as I know, there are no other Martin Chalfies in the world. So I asked ChatGPT to write a 1,000-word essay on my early childhood. It did it instantly. I have 2 brothers. It didn't mention them. It said I had a sister named Karen. Don't know why. My father never went to high school. My mother did, but it said that both of my parents were high school teachers. My father particularly was a high school teacher in biology, which would have surprised him. And it got my birthday wrong. It got everything wrong. I thought it was the best thing I'd ever seen. Because I'm not really concerned about giving students an essay assignment. Go write me an essay about X. I think the teacher should give the essay problem to ChatGPT and have it write such a completely bogus essay as was me, and give that to the students and say, fact check this. And learn about, how you can really understand the background of it. Or if you're really worried about them cheating, have them write the essay in class. So it's not a worry. But I will tell you a horrible story about ChatGPT. I have a very good friend who has 2 sons. I think the oldest is now 15 or so. And she watched her son, the oldest son, write an essay for school. So she knew he wrote the essay for school. The teacher called my friend in and said she had given an essay assignment, and because she was worried about ChatGPT writing the essays, she took all the class's essays, put them into the computer, and said to ChatGPT, did you write this? Now, these are large language models. And so the answer to any yes and no question, half the time will be yes, and half the time will be no. And so my friend was told that the teacher found that her son had used ChatGPT to write his essay, which she knew he hadn't. She then spent the entire next couple of days taking paragraphs from her son's essay, putting them into it, and half the time ChatGPT said yes. Half the time it said no. She then took her 20-year-old wedding vows that had never been online, and she asked ChatGPT, did you write this? And it said, yes, I did. So, and it's known that they hallucinate. The problem here is if people believe that what they're getting is truth out of these programs without understanding the problem. The problem was with the teacher, and it could be disastrous for the teacher, because the teacher would have said to the kid, I found you cheating. And then the kid would have said, no, I didn't do that. And then the teacher would say, not only did you cheat, you're lying to me. So it's a matter of trust. It's a matter of, so I'm not saying it's not without things to consider, but I think there's both good and other things to consider. I think that if people need, we do this all the time. You open your phone, you do a message, all of a sudden you're getting suggested words to put into it. You decide whether you're going to use them or not, or what you're going to do. Having suggestions, I don't think, is a problem. Having things written without people knowing about it. I think this is particularly a problem, I would say, when people are writing in a language that's not their original language. because I think people feel insecure, and then they will tend, as this teacher did, to believe what was written. And I think that's the problem. I think that has to be thought about. Anyway, those are my thoughts about that. Sorry to drone on and on about this.
With this, we thank you again, Professor Chalfie, for your insightful talk. And on behalf of Semmelweis University and Center for Translation Medicine, it's my honor with my colleague to hand you a certificate of appreciation for your talk and contribution for today. Thank you so much.
Thank you.